Hello everyone, my name is Taylor Rado and here is my case connection number 2. So I'm going to start by answering case number 1. So pyruvate kinase is an important enzyme for cell metabolism as it is the final step in glycolysis. Pyruvate kinase catalyzes the conversion of phosphoenolpyruvate in ADP to pyruvate in ATP and is the rate-limiting step. As in any biological process, regulation is important to ensure that proper levels of molecules are maintained in the cell. Under normal conditions, pyruvate kinase is regulated by glucose-mediated phosphorylation and allosteric regulation. Pyruvate kinase is also subject to feed-forward activation by fructose 1,6-bisphosphate. High ATP levels reduce the activity of the enzyme, meaning that in times when there are sufficient amounts of ATP in the cell, the enzyme, and subsequently glycolysis, will be halted so that the necessary energy can be produced in places where it is needed. Pyruvate kinase is also regulated by phosphorylation, as this process activates or deactivates the enzyme as well. In the fed state, pyruvate kinase is dephosphorylated and active. Conversely, in the fasted states, as glucagon levels are elevated, pyruvate kinase is phosphorylated and therefore inactivated. Clinically, a deficiency in the regulation of this enzyme can cause major biological problems and can even play a role in cancer. Of the 4 isoforms of pyruvate kinase found in mammalian tissue, so the L form in liver and kidneys, R form in erythrocytes, M1 in the muscle and brain, and M2 in the embryonic and stem cells, it's the M2 version of the pyruvate kinase that expresses the oncoprotein MYC more than any of the other isoforms, especially the M1 form. It is interesting, however, that this isoform M2 is typically found in low activity states and is ineffective at promoting glycolysis. When thinking about why this would be beneficial, it's important to note the difference in where the kinases are present. The M1 form is present in mature muscle and skeletal tissues, where the M2 isoform is found mostly in embryonic and stem cells. One possible explanation for the oncoprotein being present in M2 preferentially over the M1 isoform could be that the M2 cells are able to self-regenerate. This characteristic means that cells marked with the oncoprotein that are destroyed can be more quickly regenerated. If the cells in the M2 isoform carried the oncoprotein, they could be marked for destruction, but it would be more difficult for them to be regenerated. It is possible that the substrates from the cancer cells are passed to other metabolic processes such as glutaminolysis, or the production of glutamine, seeing as glycolysis activity is low in the M2 isoform. The excess production of glutamine in the cells could produce more nitrogen for amino acid synthesis, lipid production, and the pentose phosphate pathway, and therefore encourages the rapid growth of these cancer cells with limited activity of glycolysis. A recent paper that was published illustrated this phenomenon of glutaminolysis to support that both glycolysis and glutaminolysis are involved in tumor metabolism. This is important when considering modes of treatment for cancer by targeting not only glycolysis but glutaminolysis as well. One area of particular interest in this paper is targeting the pathway of NADPH and inhibiting the IDH enzyme. The findings of this study support that mutated IDH did limit the replicating ability of the cancer cells. So this shows that if you kind of inhibit the glutaminolysis pathway that it could have a halting effect on the proliferation of the cancer cells, which is really important in the research. So now I'm going to answer question number 2. So the uptake and regulation of glucose within the cell is very important, as this molecule serves as a fuel source for many of the metabolic pathways within the cell. The way that glucose is taken up does vary depending on the different area that it is in the body. So, and we learned a lot about this in Unit 2. The way that glucose is taken up in the liver, we'll start there, is that in the fed state, glucose uptake is insulin independent, meaning that the GLUT2 receptor is present on the plasma membrane regardless of the presence of insulin. So whether or not insulin is present or not, the GLUT2 receptor is always on the plasma membrane of these cells, which means that glucose can constantly be uptaken. In the fasted state in the liver, glucose is provided to the brain and blood and red blood cells by oxidizing free fatty acids. This process is regulated by glycogenolysis, which is the release of stored glucose, and gluconeogenesis, which is the synthesis of glucose. In the fed state, so let's talk about the skeletal tissue next. In the fed state, in the skeletal tissue, glucose is uptaken in an insulin-dependent manner. So that's different than the liver, where it's insulin-independent. So in this case, the GLUT4 receptor will only be present on the cell membrane surface of the skeletal muscle cells when insulin is present. In the fasted state, skeletal muscle also oxidizes free fatty acids as an alternative fuel source to the glucose. So next we'll talk about the brain. So metabolism in the brain is an aerobic process, and glucose is taken up in an insulin-independent manner by the GLUT1 and GLUT3 receptors. Glycolysis in the brain will always oxidize glucose except in cases of extreme starvation. In the fasted state, glucose will still be taken up by the GLUT1 and GLUT3 receptors and will be oxidized through glycolysis. So the last part of the body we'll look at in how it uptakes glucose is the adipose tissue. So in the fed state, in the adipose tissue, glucose is taken up in an insulin-dependent manner by the GLUT4 receptor. In the fasted state, free fatty acids are provided through lipolysis so that the stored free fatty acids can be oxidized by the liver and by the skeletal muscles. So if we look more specifically at the case study shown in our example for number 2, and the MRI scan showed to the left, it's clear that the area of the brain where the tumor is located has an increased amount of glucose uptake. So the one on the left, you can see the tumor kind of outlined in the white, and then on the right, you can see that the glucose is mostly in the same area as the tumor. So in the left image, the white area outlines the tumor, and when the D-glucose is fluorescently labeled in the left image, in the right image, the glucose is built up in the same area the tumor is outlined. This would suggest that the tumor requires a certain level of glucose in order to grow. In line with our knowledge of glucose as a source of fuel for many metabolic pathways, this would make sense. Glucose is used to make ATP and other molecules that help cells to grow and survive, and therefore would be necessary for the tumor cells to stay active and to proliferate quickly, as they do in most cancers. Unlike normal brains, where the glucose uptake may be more uniform and spread out throughout the entirety of the brain, glucose uptake seems to be consolidated to the area of the tumor, as if those cells are almost hoarding all of the excess glucose. A possible explanation for this is that the cancerous tumor cells could be expressing and upregulating more GLUT1 and GLUT3 receptors on their plasma membranes. The more receptors that these cancer cells are expressing or upregulating, the more glucose that they can uptake into the cells, causing for this increased level of glucose in that area of the brain where the tumor is. It is also possible that these cancer cells have a mutation in the regulatory enzyme pyruvate kinase. If this enzyme isn't regulating the amount of glucose that is coming into the cell and allosterically shutting off the intake, then the cells will continue to uptake glucose even if it is not necessary for fuels in these cells. Without this enzyme functioning properly, glucose levels can climb to abnormally high levels without any regulation. Thank you for listening, and I have my sources cited in the attached document. Thank you.